meeting, and I realize that for some of us, bandwidth may cause some issues for us while we are conducting this webinar. So I would ask, please, if you could keep your video and your microphone off for the bulk of the presentation. And also, can I ask for questions that you might have, it's probably going to be easiest if you actually put them in the chat function. So the chat function, if you're new to Teams, is a nice little messaging board in the toolbar. And at various stages in the afternoon, my colleague, Dr. Jaime Portelas, will be asking me questions. So I've broken the presentation up into 4 sections, and at the end of each of the 4 sections, there will be a conclusion slide, and I will break at that point to respond to the questions, and I'll ask Jaime to read out questions at that point. If you feel that he's not captured your question satisfactorily, please, there's a raise your hand button within the toolbar of Teams, and so there will be the opportunity to interact more personally over the course of this webinar. So I think, having covered pretty much all the housekeeping arrangements, I'll now present our webinar, and the title that I've given for today is Partial Root Zone Drying from Laboratory to Commercial Practice. And hopefully you can all see the welcome slide. Can I just have affirmation from someone that I've successfully shared my screen?
Yeah, you're right.
No longer seeing me.
Yeah,
is that
right. Correct?
Yes.
Fantastic. So today's webinar is part of a much larger project, the SHui project, which is funded by the European Union. And you can see from the title of our project, Managing Water Scarcity in European and Chinese Cropping Systems, that it is very much a joint effort. And we're looking at a range of different soil management and water management techniques to try and improve the productivity of various cropping systems in both the EU and China. Now, I would encourage you to have a look at our website to learn a little bit more about the project. But if you're academically minded, we have actually recently published an article in the International Journal of Soil and Water Conservation Research. So I would direct you to that. As I mentioned, today's webinar is part of a series, and in some ways we're going in reverse chronological order. So our colleagues in China hosted the first webinar 2 weeks ago, and so today the topic I'll address is partial root zone drying from laboratory to commercial practice. If you're interested in some of the other titles that I've put up within this series of webinars, please get in touch with us. We can send details to you. At the moment, the delivery dates are categorized by month, but very shortly they will be categorized more formally close to the time by date. So if there's something of interest for you there, please get in touch with us via the SHui website, where you can contact us. So the topic, as I've indicated, is partial root zone drying. And what I want to do is consider how this particular technique has evolved from both scientific but also commercial purposes. Now, the European Union that sponsors the SHui project has this sort of linear gradient of technology development. You know, starting with perhaps a bright idea in academia and via collaborating with appropriate partners, ultimately makes its way into the industry or commercial world. And of course, there are a lot of interchanges and interactions between partners on the way, between idea through to commercial reality. And hopefully I will capture some of those in the course of the webinar this afternoon. In terms of partial root zone drying, I think it's relatively easy to sort of see a route to develop. in the sense that for some of us that are on the call, partial root zone drying is a convenient technique that we could apply hot trials or controlled environment or CE trials. Then hopefully we have the opportunity to do field experiments to evaluate this particular irrigation technique, and then hopefully commercial partners may be interested in applying this. Just can I ask again for those that might have joined the webinar recently, if you could turn your microphones off, that would be very much appreciated, because I'm getting quite a lot of background noise at the moment. Anyway, I will launch into my discussion of partial root zone drying, and the story starts for me before I was a scientist, and the story starts here in Lancaster in the United Kingdom. Now, Lancaster might seem a very strange place to start a story on partial root zone drying. If I look outside my window at the moment, it is raining heavily, and certainly you wouldn't argue that water scarcity for agricultural purposes is likely to be a big problem in the northwest of England. However, I think the story on partial root zone drying probably starts with some work from Professor Bill Davis, who's actually on the call here today, working with one of his PhD students, Peter Blackman, in the 1980s. They were interested in how plants actually respond to soil drying. And in this case, they were very concerned with growing plants in pots, that it would be, if the plants were transpiring relatively quickly and the plants were grown in quite small pots, that very rapidly the plant would go from a situation where it was well watered to very dry, potentially suffering a lethal water deficit simply because of the large plant size growing in a small pot. Now, if they were interested in the impacts of drying soil distinct from the impacts of plant-water relations, it was necessary to try and slow down the rate of drying soil. And in this case, there was a fairly ingenious split root system developed. The initial experiments were with maize plants with their root systems grown between two pots. And this experiment tried to ask the question whether the plant response was a response to changes in leaf-water relations or a response to changes in the soil environment affecting the biochemistry occurring within the root systems. And this is a relatively old paper now, somewhat of a citation classic, and I think it's a nice starting point for our scientific investigation initially of partial root zone drying. So these are the results of the partial root zone drying experiment. As you can see, there were two treatments, some well-watered plants that were irrigated on both sides of the pot. And here are our plants exposed in this split root system to partial root zone drying, where part of the root system was well watered and the other part was exposed to drying soil. Now, Peter and Bill made some very careful measurements of the water relations of the plants, and you can see here that if anything, the plants exposed to the partial root zone drying had a slightly higher turgor as the soil dried over the course of this experiment. These are the component measurements of leaf water potential and osmotic potential. Now, at the time, the prevailing wisdom in terms of plant-water relations was that plant responses to drying soil was simply a function of the water relations of the leaves. So we might expect, with very limited changes in water potential and osmotic potential and turgor of the leaves, that actually these plants exposed to partial root zone drying would show no significant physiological response. But yet that wasn't the case. These are some data from a viscous flow pyrometer. Unfortunately, I was unable to secure a picture of a viscous flow pyrometer from the internet, so I've included a transient time pyrometer, which I suspect many of you will be familiar with. But what this allowed the researchers to do was actually to look at a diurnal course of stomatal conductance. And you can see here for the well-watered plants that initially when the lights came on in the controlled environment cabinet in the morning, the leaf conductance substantially increased, and that was maintained throughout the course of the day. For the plants exposed to partial root zone drying, the stomata opened in the morning, maintained reasonably high stomatal conductance for the first few hours, but then slowly closed and reached a lower steady state value over the course of the rest of the day. So we can see here in this case that stomatal conductance of the plants exposed to partial root zone drying decreased. And it's very difficult to argue that this was a direct response to a leaf water deficit, because if we go back to our previous slide, we can see that if anything, the turgor of these plants exposed to partial root zone drying was in fact higher. In fact, what I would argue, and the authors of the paper argued, was that actually this stomatal closure was necessary to maintain the turgor of the leaves of the plants exposed to partial root zone drying. The sort of key leap here was the hypothesis was that the roots that were in the drying soil were producing chemical signals that were moving from the roots to the shoots to actually effect the stomatal closure that occurred. Now, in this study, there were some measurements made of a particular plant hormone, abscisic acid, or ABA, and I will be talking a lot about ABA or abscisic acid over the course of this afternoon. I will try and keep some level of moderation on my enthusiasm. And actually in this study, it was difficult to demonstrate that ABA was actually increased in response to the partial root zone drying. But hold that thought for the moment, because it's a point I want to come back to. In a later experiment done with partial root zone drying in the same laboratory, David Going was working with apple seedlings. And in this case, the partial root zone drying plants showed a restriction in leaf growth as the soil dried over a much longer period of time, in this case up to 24 days. Now, again, this was interpreted as evidence that the leaf growth was being constrained by chemical signals moving from the roots to the shoots, again, because the measurements of the leaf-water relations did not show any significant change between the two groups of plants. And within this experiment, there was also a sort of recovery phase included in the experiment. Now, this is the recovery phase of the experiment here. So here we have the well-watered plants that received irrigation on both sides of the root zone, and they maintained a high leaf growth rate. This bar here represents the plants that were continued to be exposed to partial root zone drying. And then we have two treatments applied here. So in one case, the partial root zone drying plants were re-irrigated, and you can see that the growth rate of the leaves recovered. In the other case, the roots in drying soil were excised, and again the growth rate of the leaves recovered, again demonstrating that it was probably chemical signals moving from the roots to the shoots that were actually controlling the responses of these plants. So these series of split root experiments that had been done in Bill's laboratory attracted the attention of a group in Adelaide at CSIRO Plant Industry, the group led by Brian Loveys. And at the time, they were interested in the problem they were having with some grapevine cultivars showing excessive vigour under their conditions in Australia. And so they did a very similar split root experiment with either whole root zone drying indicated here or partial root zone drying indicated here. Now these, again, this blue bar represents the plants that were well watered, and you can see when we compare the stomatal conductance of the plants exposed to whole root zone drying, that the stomatal conductance crashed relatively quickly, and then the plants were re-watered at this point here, and then the stomatal conductance increased again over time. For the plants exposed to partial root zone drying, there was a slightly slower or delayed stomatal response. So you can see that the stomata closed, and then even though the soil continued to dry on the dry part of the root system, there was some stomatal opening occurring, as we can see here. It was only at this point here that the roots were re-watered. And so this is quite an interesting response, so that we can see that the stomata close, yet with continued soil drying, the stomata start to reopen again. And this is, I think, a key observation that resulted in a change in practice. So the thinking was that if the researchers wanted to maintain this stomatal closure over the course of a prolonged period of time, it was actually necessary to swap or alternate the dry and the wet parts of the root system. And this symbol here that you can see represents the swapping of the wetting and drying parts of the root system. And this alternation of the wetting and drying parts of the root system, as it's known, has been a sort of fundamental part of this more commercial or field application of partial root zone drying. So some conclusions from this first necessarily rather academic, and I would say in terms of technology readiness level 1, so the discovery phase of the research. So the sort of key conclusions, leaf-water relations don't always regulate the shoot response. We've seen a couple of examples in split-root maize plants and split-root apple seedlings, where the water relations of the plants are maintained as part of the soil dries, and yet the stomata closed and the leaf growth was restricted. So the argument or the explanation for this particular behavior of the plants is that the roots are detecting the drying soil and actually sending signals to the shoot. And I've mentioned one of these potential signals, ABA or abscisic acid, and that's a hormone that I'll come back to later in the presentation this afternoon. And these signals seem to be remarkably effective in closing the stomata and actually restricting the growth of the leaves. So I'll pause my screen sharing at this point, and I will see whether there's any questions at this point. Jaime, did you have any questions that have come in on the chat?
So far, there's no questions. So if you want to ask any question at the moment, please do it now, or we continue.
So please do ask questions if you have them when I get to one of these conclusion slides. I know over the course of the first part of the presentation that I've heard lots of people joining the presentation. I can hear some traffic noise in the background. Yeah. So that if you're new to the webinar, can I just ask that if you could mute your microphone, that would be very helpful. And also to note that our webinar is being recorded today, because ultimately we want to place it on our website as a record of activities from this particular project. However, at the same time, I hope that doesn't inhibit your questions and your enthusiasm for this particular work. So if there's any questions, I'll take them now. Otherwise, I will move on to the next part of the presentation.
So Ian, there is a comment from Bill Davis. And he's commenting on in the study from Gowen, much of the increased root growth after cutting dry roots was the production of new leaves. So that's a remark from Bill.
Yeah, I'll go back to that slide I've taken from the paper that David Going produced, and obviously Bill will remember it better than I, because I only read it, whereas Bill actually participated in the study. So here the data is expressed as a growth increment, and I believe that growth increment is a leaf area increase.
Yeah, he said that he meant leaf growth, not root growth. Yeah.
Yes. So it's leaf growth, and that's a leaf area increment, and of course that can be driven by two processes. So that the leaves can expand that are already on the plant. So this is leaf area expansion. But also in this particular study, there was some very careful measurements made of leaf number. And in this case, the partial root zone drying had a very significant impact in actually reducing the initiation of leaves as well. So at least in apple trees, the response to partial root zone drying seemed to not only be a slowing of the leaf expansion, but also a restriction of leaf initiation. When the dry roots were excised, indicated by the scissors here, or re-watered, there was an increment in leaf area again, and the plants sort of returned to almost a well-watered state. And this was driven by increased numbers of leaves and increased leaf area. So I hope that addresses the comment. Other questions or comments at this point?
Not at this point.
Not at this point. Okay, so I want to move out of the laboratory now and into the field. And as I mentioned, the group in Adelaide at CSIRO Plant Industry were concerned about the levels of water use, but also excessive vegetative vigor of some of the grapevines that were being grown in the Barossa Valley near Adelaide. And so they were aware of the work that Bill and his team had done on these split root experiments and wondered whether these could be adapted to a field environment. So what they set out to do was to establish a split root system in a vineyard. Now, initially, the experiment used real vines, and a vineyard was planted with a plastic sheeting down the middle of the row to actually physically separate the two parts of the root system. So a very similar system to what had been applied within the potted maize plants or the potted apple seedlings, but actually in a field crop, in this case, grapevine. And so the technique was implemented essentially as it had been done in the controlled environment experiments, and the wetting and drying parts of the root system were regularly alternated over the course of a growing season. Now, this particular technique was applied during the PhD studies of this gentleman here. This is the rather aptly named Peter Dry, who was working with Brian Loveys at the time. And this is another of the students who was involved in the work. And this is one of their original vineyards with partial root zone drying. And you can see they've implemented the partial root zone drying by applying 2 drip tapes on either side of the row of vines. And this particular technique that they called partial root zone drying was promoted quite widely to vineyard owners and growers within this part of Australia. And in fact, this is an image I've taken from the Powerhouse Museum archives, Powerhouse Museum being a well-known museum in Sydney. So here, and what it has done is tried to capture various innovations that have been applied within Australian agriculture, and this is one of them. Now, for the benefit of the students in the audience today, you'll probably always be told, don't go to Wikipedia for your information for your research papers. In this case, I'm going to ignore that particular advice, because if you look on Wikipedia or Wikimedia, and I get the two confused sometimes, there is actually a rather pixelated diagram of partial root zone drying that you can find. But I think it explains quite nicely the concept, so that you have roots that are in drying soil that send these chemical signals to the shoot, triggering various responses. And then you have another part of the root system that is well irrigated, and these irrigated roots continue to provide enough water and nutrients to the shoot to allow it to function. So this partial root zone drying technique has been popularized very much in Australia in vineyards, and you can see here an example of how it might be applied with these drip tapes and the different wetting and drying parts of the root system in different vines as you move along the row of the vine. So for the initial irrigation cycle, you can see you've got a dry part of the root system on this vine, a dry part of the root system here, and a dry part of the root system here. And then these parts of the root system here have been irrigated. After a period of time, the wetting and drying parts of the root system are alternated, and you can see that the dry side is then re-irrigated, and then the irrigated side is allowed to dry the soil. And the reason for this alternation was to ensure that these chemical signals continue to move from the root system up to the shoot to regulate the stomatal responses, but also control the vigor of the grapevine. So what were the results of these particular experiments like? And these are some of the original data from Brian and Peter's experiments with partial root zone drying. And the results seemed, relatively positive, and there was a lot of industry interest at the time. So that you can see by applying the partial root zone drying over a period of time during the season, and you can see that, it was an alternate partial root zone drying that was applied, there was a considerable reduction in the amount of irrigation that was applied. There was also, despite this, relatively little change in the weight of the fruit that was picked between the two different treatments. Probably not even statistically significant. So this was a very positive result as far as the industry was concerned, because the amount of water had been restricted, yet the vines continued to yield relatively normally. And the reason the vines managed to achieve this was because they were less vigorous, and so had put more resources into the weight of the fruit. So you can see a reduction in leaf area, a reduction in pruning weight, but also a reduction in stomatal conductance. And so by reducing leaf area and reducing stomatal conductance by applying the partial root zone drying, the plants actually used less water, and this is probably how this reduction in irrigation, managed to be achieved. Now, of course, for those that love wine out there, you would recognize that it's more than just about determining the amount of yield of the berries. The quality of those berries is absolutely critical to determining the quality of the wine that is actually produced. And again, these are some of the original data that Brian has provided for a couple of vintages from last century, I would add, 1997, 1998, measuring some of the chemical changes in the berries that had been exposed to partial root zone drying. So you can see here a number of different chemical components that were increased within the plants exposed to partial root zone drying, particularly from the 1998 vintage. So obviously a very good year for for partial root zone drying wine production that year. And I've got here a sort of summary statement that quality can be very important in certain crops, and certainly for wine grapes they are one of those. And that more generally, independently of how you apply less water to the root zone, whether you adopt partial root zone drying or conventional deficit irrigation, where you apply less water to the entire root zone, this concept of deficit irrigation can be useful to increase specific chemicals in the berries and therefore impart certain flavours and aromas to the constituent wine that is produced. So as I mentioned, there was quite a bit of industry interest within Australia at the time in adopting this partial root zone drying technique. Of course, there was also, the communication of these results at various conferences around the world. And certainly for a conference that I've attended and some of my group has attended is an International Society of Horticultural Science symposium that is run by their irrigation group. And certainly when these results were presented, they attracted quite a degree of interest and probably a healthy degree of skepticism. And when I teach these results to my third year students, I always make the point that this particular experiment's a lovely demonstration of what can be applied in practice, particularly based on some of the scientific theory that I covered in the first part of the presentation. But actually, in terms of an experimental design, this particular experiment has its limitations, and the limitations are that in this case, not only have you changed the irrigation volume with the partial root zone drying plants, but also you've changed where the irrigation is being applied to the root system. So if you think about it in a sort of experimental design format, we've got a confounding within the partial root zone drying of changes in irrigation volume and irrigation placement. And in fact, the irrigation science community was quick to pick up on this. And there was some discussion of this particular technique at one of these irrigation symposiums. I should just say that Oxford Landing was one of the vignerons in Australia that successfully adopted this technique within part of their vineyard, and certainly I was fortunate enough to actually visit to discuss with them how they were applying this technique. But I'll return to Oxford Landing perhaps again later in the presentation when I consider the commercial application of this partial root zone drying technique. So as I mentioned, there was quite some discussion of this partial root zone drying technique at an irrigation symposium. And I've sort of posed the question, well, what did the irrigation science community think? And the reality was, well, there was a healthy degree of skepticism. In fact, you might even say outright skepticism and possibly even condemnation. And certainly this was a post on this website that at this symposium on the irrigation of horticultural crops, the conclusion was that PRD does not offer any benefit over conventional irrigation. And it was the statement was made, and these blogs are always great for allowing people to articulate exactly what they mean. That if something doesn't work as advertised, I think we need to be very diligent about letting people know it doesn't work, especially if it was advertised heavily. And I think this is an important message for all of us that actually seek to sort of go from science into the commercial world. That often in the commercial world, there is the expectation that the technique is always going to deliver exactly what the proponents of the technique might say it does. And I think that's probably something that scientists recognize very well. And we can see here in the analysis of the quality of the grape berries, very considerable year-to-year variation in the levels of these particular chemicals within the grape berries. So context dependency is something that scientists understand very well, but it's probably not something that the commercial world it wants to hear when we are trying to advocate an agricultural technology. And some more comments about this partial root zone drying technique that certainly for me as a scientist was very interesting, that there is nothing magic about alternating the wetting and drying sides, and that the responses are essentially just response to moderate water deficits. And so I'm going to return to these particular 2 points that appeared on this irrigation forum as we go through the presentation. A very nice statement that or succinct statement from Ken Shackle. As far as I can know, PRD is DOA. Succinct in the sense that it uses acronyms, and I would always advise my students that if you're going to use acronyms, make sure you define it. And indeed, this was seized upon in the next post on this irrigation forum that said that DOA is a TLA for dead on arrival. And for those that don't know what a TLA is, I would say it is a 3-letter acronym. And again, this is something that I always talk to my students about, that it's very important to define the acronym. The other point I would make in thinking about this discussion that I would argue is, scientists sort of having a drink and, discussing things fairly informally, that it can have a sort of negative impact in terms of grower uptake, or allowing growers to make their own mind up about a particular technique. In terms of the academic world, partial root zone drying is something that has captured our attention. So if I do a search in Web of Knowledge and search for partial root zone drying, I find a large number of papers over the preceding 20 years. So if I look at that discussion that was in 2003 about whether partial root zone drying was dead on arrival, well, you could say that that didn't stop a huge number of people publishing the experiments that they'd already done. Perhaps since 2007, there has been a declining interest in partial root zone drying. Perhaps. For me, who read this discussion in 2003 and thought, well, I don't want to work on partial root zone drying for much more in my life if it is actually dead on arrival. For me, this sort of sustained interest in the technique has been very heartening. And indeed, this comment, dead on arrival, for me was very much a red rag to a bull, if you'll pardon the expression, because it stimulated me to think, well, how do we actually understand the science behind this technique a little better? I mentioned that partial root zone drying has been applied in the field by scientists throughout the world. And each one of these stars represents a country where I've managed to track down a paper that has actually applied partial root zone drying in the field. Now, I realize that some of you in the audience are probably the originators of those stars. You may have trials that you have done previously. You may have trials that are ongoing. And so it's your efforts that have allowed me to populate this map of the world in terms of understanding the global interest by academics in this particular technique of partial root zone drying. So I applaud you for your efforts. If you're on the call and there's not a star in your particular country, please get in touch with me after the call and let me know about the work that you or your colleagues have been doing. This map does evolve over time, and it is completely possible that I haven't captured some of the more recent experiments that have been done in the field with partial root zone drying. This sort of series of maps and crops just indicates a bit of a snapshot of some of the different crops that partial root zone drying has been applied to within particular countries around the world. It is just a snapshot. I predict that I could put up probably 10 or 20 slides of a similar nature, because I understand there's been, several hundred publications that have evaluated partial root zone drying in the field in relevant environments. I want to turn to another snapshot, and this relates to the comment that was made in the irrigation discussion as to whether partial root zone drying was just a response to moderate water deficit. So this is a snapshot of 15 studies that I compiled more than 10 years ago. It comprises field studies that are indicated by the hollow bars, and the pot studies, which are indicated by the shaded bars. Now, in this particular slide, the two groups of plants, those that were exposed to conventional deficit irrigation, and you can see here that most of the soil is partially dry. The deficit irrigated plants and the partial root zone drying plants receive the same irrigation volumes. Now, what I've done within this particular slide is I've calculated the yield ratio of the partial root zone drying plants compared to the deficit irrigated plants. So if there's no difference in crop yield between this group of plants and this group of plants, all these studies should result in bars that lie on this line here, which is 1. So a ratio of 1 indicates no yield difference between the two treatments. Now, certainly you can see examples within this particular slide where the studies have shown no difference between the two techniques, or indeed in one case actually a slight reduction in yield with the partial root zone drying. For many of these studies, the partial root zone drying significantly increased the yield compared to the deficit irrigation, and if there's an asterisk on the bar, that means it was statistically significant. Now, at about the same time, a much more substantial meta-analysis appeared in the literature by Victor Sadras, who'd been working in CSIRO in Adelaide, comparing partial root zone drying with deficit irrigated plants. And we've got studies from different grapevine varieties at the bottom, but also a whole range of perennial tree crops here. Here we have the yield of the deficit irrigated plants and the yield of the plants exposed to partial root zone drying. And you can see that there's a linear relationship between the two, as you might expect. And in some cases, the partial root zone drying plants appear above the line, indicating that they have enhanced yield compared to those that were exposed to deficit irrigation. So across all the studies, you can see this sort of summary graph at the top, looking at the percentile and the percentile difference. And so we can see that there are relatively few studies that show partial root zone drying results in more than 20% yield increments. So about 20% of the studies show more than 20% yield increment. Now, you can argue whether a 20% yield increment is worth having. I suspect for most farmers, they would be glad to receive this. Many farmers, in fact, might be happy just to increase yield by 10 to 15%. If we look at the original snapshot of the data that I presented, again, almost all the statistically significant results, there was a 20% yield increment. Now, more recently, Michael Adu, working at the University of Cape Coast in Ghana, has done a couple of meta-analyses compiling the data since the studies that were done in 2009, comparing partial root zone drying versus deficit irrigation. And this is the consensus response, a slight increment in yield, but obviously considerable variability across the different studies done with field crops, vegetables, tree fruits, and vine crops. He followed up this yield analysis with an analysis of quality, so looking at the soluble sugars of the crops that were harvested, and again, a slight increment with partial root zone drying, but substantial variability across the different groups of plants that were considered. So the technique of partial root zone drying has been agronomically evaluated very extensively over the 20 years or so since it has been introduced. There's been a huge number of trials done predominantly by academics, collaborating often with commercial partners in field environments. And I would argue that these are the sort of conclusions from the work. Certainly, it's been possible to apply these split root systems in field trials. Now, the group in Adelaide very quickly established that you didn't need to actually put a physical divider down the middle of the row of grapevines to ensure that you had spatial separation of wetting and drying parts of the root system. At least in the very sandy soils that they were working in Australia, it was relatively easy to put 2 drip tapes down the side of the row of vines and have distinct wetting and drying parts of the root system. In clay soils, this is not so easy to achieve, and certainly I can remember speaking to cotton researchers in Western Queensland that had tried to apply partial root zone drying in their very clay-rich soils. These very clay-rich cracking soils that crack up when they dry, it was almost impossible to establish distinct wet and dry parts of the root system. When the dry soil started to crack and then was re-irrigated, the water just went everywhere and the whole root zone became re-irrigated. So certainly within certain soil types, it's relatively easy to establish these split root systems in field trials. The recommendation was to alternate the wet and dry parts of the root system to try and maintain this chemical signaling from the roots to the shoots. I'm going to talk a little bit more about that in the next segment of the presentation. And certainly there was some evidence of enhanced yields in some studies, but this couldn't be guaranteed. And I want you to store that thought when I come to the commercial application of partial root zone drying in the final part of the talk. So at this point, I'm going to indulge in some irrigation myself. I need a drink to give my voice a rest, and hopefully Jaime has got some questions stored up for me.
Yeah, I've got a couple of questions, so I let you Ian to irrigate yourself, and we can go with them. So all the questions come from Sven Brookfeld, who is a commercial grower, a grapevine grower in Chile. Works with Pilar Gil from Universidad Católica de Chile, and he's interested in some questions. The first question is, what if leaf area goes down and then yield goes up? What about the long-term sustainability for the vine? So he talked about, I don't know, I'm not an expert in grapevines, so I don't know whether it's the Rabat index, but he said that the Rabat index will change.
Yeah, so certainly longevity is an issue that any commercial grower needs to consider over the course of the cropping cycle. I mean, I'm not a grapevine expert, but I would imagine that a grower would expect grapevines to last 20, 30 years and be productive during this period of time, and certainly if vigor is reduced in 1 year, there may be a concern that in subsequent years there is insufficient material to generate the vegetative growth in the following season. However, even the plants exposed to partial root zone drying in the original experiments, there was some need for pruning even in those plants. Of course, one of the challenges that scientists often face is that often our funding cycle comes in sort of 3 or at most 5 years. Now, I am aware with some people managing to get funding for longer term experiments, that partial root zone drying has now been evaluated not just in 1 or 2 seasons, but up to 5 years or more over a prolonged period of time. And certainly it's these longer term experiments that I think provide some comfort for growers that these techniques are actually sustainable over the longer term. And I hope that answers the question. And I would certainly be interested to hear from others on the call that are working with grapevine as to what their experience has been over the duration of experiments that they have implemented.
Okay, so there's another question from Sven, and I think this is, for me, one of the million-dollar questions about PRD. What is the good timing for changing from dry to wet area? And I think this is something that I personally am working on that at the moment. So and that's something that probably brought me to Lancaster in first place. So I'm very interested to hear from you, Ian, again, about what is your opinion about this.
Yeah, well, feel free to answer the question yourself, Jaime, but from a sort of theoretical perspective, the alternation of the wetting and drying parts of the root system should occur at the point that the stomatal closure response starts to reverse. The stomata start to reopen because the roots in drying soil are contributing less signal from the roots to the shoots. Now, you would argue that that is a very academic answer, and it may be very difficult to actually establish this in any field system. And the reason for that is that within the field system, not only are the roots laterally spread so that there are roots in wet and dry soil, but there's also substantial vertical gradients in soil moisture. Now, if the soil is relatively deep, you might get the situation that actually, you never get this reversal of the stomatal closure response, because always as the soil on the dry side of the root system starts to decrease, it will just decrease not only in amount but also in depth. So in a very deep soil that might extend to 1 to 2 meters, you may never need to actually alternate the wetting and drying parts of the root system if some of the roots in the dry soil, if there are roots in the dry soil that are starting to dry out at depth and the chemical signal is maintained throughout that drying period. In practical terms, that's not a satisfactory answer. And certainly I would argue that most of the experiments that researchers have done around the world have been very empirical when it comes to wetting and alternating the wetting and drying parts of the root system. So in the original work done by the group in Adelaide, 2 weeks was about the period that they settled on, and that alternation duration has been to a large extent adopted by the research community over time, and I would argue with a surprising lack of spirit of inquiry as to whether 7 days, 14 days, 21 days or 28 days is, or even longer, is appropriate within a field crop. I know, or I hope, Juan Gabriel Perez from IVIA in Valencia is on the call, and I know he's had some very positive results with actually in citrus with substantially extending the period of time between alternating the wetting and drying parts of the root system. I've probably said enough at this point, so I've reverted, I think, to type in terms of an academic answer to the question, without necessarily thinking of the practicalities of determining the ability to distinguish any sort of stomatal reopening response as a result of prolonged drying of part of the root zone. Jaime, I don't know whether you want to comment at this point, because I know you have thought about this quite a bit as well.
Yeah, I think it's quite complicated, and I think there are many factors that determine the timing. One possibility would be to focus or to see or to have soil moisture sensors and determine when is the right time, depending on the soil moisture, the dry side, how much we want to allow the soil to dry in the dry side. But there is no empirical evidence, or there is no evidence what is this right time, and it depends very much on the species and also the objective of that. So I think that's still a very open question, and it's one of the research areas that PRD still offers.
It's not a very reassuring answer, I assure you, but at the same time, it suggests that there is much more that we need to do. And I would argue that it's this complexity of when to alternate that might have actually inhibited the adoption of the technique, because the optimal duration of alternation will vary according to soil type, according to evaporative demand of the atmosphere. I mentioned Oxford Landing earlier in the presentation, and when I talked to the vineyard manager about the block that they had under partial root zone drying, his response was, "Well, we alternate on a Monday." And I asked, "Well, why do you alternate on a Monday?" And he said, "Well, if we don't do it at the start of the week, we're liable to forget." So complete empiricism in terms of that answer. But it worked for them, and they were very happy with the application of the technique on that particular block of their vineyard.
Yeah, regarding that empirical empirical way to swap, I remember also the comment from, I think, from Jianhua Zhang about how they decide to swap sites in maize in China. It's basically when you can see your footprint. When the farmer goes to the field and he cannot see the footprint in the dry side, it's time to irrigate and swap to the other side. So, but it's not a very scientific answer, but that's what we have so far. Ian, we have quite a lot of questions, so maybe we can go on and continue asking you those questions.
That's fine. Yes.
So okay.
The next part is unapologetically scientific. So I'm very happy to take questions at this point.
Okay. So another one from Sven. What about the effect of slopes? How more difficult it is? How this complicate things? Having to apply PRD on a slope.
I've seen your vineyard, Sven, on the internet, and yes, there will be challenges, I suspect, of applying partial root zone drying on slopes, and I'd be keen to talk to you offline about what those are. I mean, the sort of assumption with the sort of level vineyards is that when you apply drip irrigation, you've got a wetting bulb that is spreading vertically rather than laterally down the slope. So there may be problems with actually ensuring distinct wetting and drying bulbs of the root system. I would expect that if it's a drip irrigated system, we're not too concerned with potential problems of soil erosion down the slope.
Okay, so another question, this time from Bill Davis, who also makes a very, very interesting point. So when we are comparing depth irrigation, homogeneous depth irrigation and partial root zone drying, whether or not there is a difference in water use efficiency, in the ratio between yield and water application. How important is that both treatments are applied with precision, or how common is that we have very good studies in which they apply with sufficient precision the techniques in terms probably, well, I suppose in terms of measuring the water and applying the irrigation in an appropriate manner.
Yeah, certainly scientific precision in all of these experiments is absolutely critical. For me, I think the greatest limitation in some of the science is just accurately understanding, following the application of water to different parts of the root system, where that water actually ends up. Soil moisture sensors, usually only measure a relatively restricted sort of volume of soil around the sensor, and so it can become quite challenging, especially with partial root zone drying experiments applied at scale, to actually satisfactorily monitor whether you have got discrete separation of wetting and drying parts of the root system. And I would argue that spatial separation, particularly within more clay-rich soils, can be particularly difficult to achieve. And when I was working with plants in pot environments, I did an analysis trying to understand whether the difference in soil moisture between the wetting and drying parts of the root system was actually very important in determining whether I could distinguish a distinctive partial root zone drying effect or not. And that seemed to be the case, that if you apply, even regardless of how careful your application of partial root zone drying is, if you're not achieving the distinct wet and dry parts of the root system, for example, you have more roots in one part of the soil than the other, it's very difficult to substantially satisfy yourself that you are actually getting a distinctive partial root zone drying effect.
So another question this time from Michelle Ganem. How PRD, or are you aware of any studies on how PRD affects the relationships between roots and microorganisms?
There's a little bit of work that Shanhua Zhang's group in China has done looking at just the number of culturable microorganisms as a function of soil moisture content in plants exposed to deficit irrigation and plants exposed to partial root zone drying. And at an intermediate water content, there seemed to be a higher level of culturable microorganisms for the plants exposed to partial root zone drying than plants exposed to deficit irrigation. I think the mechanistic significance of this is a little bit more difficult to understand, given that culturable microorganisms is a very sort of crude measure of the sort of physiological function of those organisms within the soil. But I'm not aware of much information on how these different irrigation techniques affect plant microbe interactions. And potentially that can be quite important in determining the response. So certainly soil scientists are very well aware of something called the Birch effect, where if you re-water dry soil, you get a pulse of nitrogen liberated from the soil. Phil Haygarth here at Lancaster University has worked intensively on phosphorus release from soil that has been dry and then re-wet. And these effects could be quite important in actually improving the nutrition of plants exposed to partial root zone drying, if, and I think there's a big if, the roots can actually access these nutrients that might be liberated from the microbes, and if rather than other soil microbes actually taking up those nutrients themselves. So again, here is an area that I think hasn't been explored in sufficient detail. I know Fulai Liu and his group has done quite a lot of work on crop nutrition under partial root zone drying, and I would refer you to some of the articles of Fulai's group.
Yeah, thanks, Ian. So now a question from Chet and Panda. So how different is training, pruning, thinning, like chemical thinning with low regulators like ENAA in a PRD system compared to a system in which you use complete irrigation, full irrigation?
Yeah, I'm probably not best qualified to answer this, having not worked with grapevines myself. I would refer back to the slide about the pruning weight of the plants exposed to partial root zone drying being less. Whether that actually results in changes in thinning or the application of growth regulators, I am less certain of, and I suspect there are many on the call that have worked with grapevines that are perhaps better qualified to answer that question than I.
Okay, so now another question from Paula Serrano. Are you aware of any studies on how PRD affects Phytophthora disease or inoculum in soils or its relationship with biotic stress?
So when I was fortunate enough to visit Malaga in Spain more years ago than I care to remember, I did have a conversation with a group there that were applying partial root zone drying explicitly to try and decrease levels of Phytophthora inoculum in avocado. Now they were very positive about the effects of the partial root zone drying in reducing Phytophthora levels. I've yet to see those results being published. That's not to say that they haven't been published, perhaps in the Spanish agricultural journals, but I haven't actually seen those. But it certainly was regarded as one of the benefits of applying partial root zone drying in the avocado crop. And there may be other examples worldwide where the technique has been used similarly. Again, potentially an unexploited area of research in coming back to Michelle's comments about microorganisms and plant interactions.
Yeah, another question from Juan Gabriel Perez. What do you think about the use of PRD with low quality irrigation water?
So I think there's been quite a lot of work done by CSIRO plant industry on this. The concern, of course, is that with applying less water, you'll get a concentration of salts within the root zone. I think here we are limited very much by the funding cycle. I have seen reports out of some of the Australian groups that there was no toxic effects of applying less irrigation water, either via deficit irrigation or partial root zone drying. But I suspect in those systems there was some substantial winter rainfall that might have modulated any impact of salt accumulation over the course of the irrigation season. So certainly I would argue that this is a concern for anyone who's interested in applying low quality water via deficit irrigation or partial root zone drying. One thing that captures my attention is with partial root zone drying, if you're applying the same volume of water to only part of the soil, you should get greater leaching of salts through that part of the soil compared to the same water being distributed over a much greater area of the root zone. But certainly this is something that I think needs a little bit more work and considerable care and attention in applying any form of deficit irrigation with low quality water. And often there are concerns that whereas the results that may be relatively positive for the first couple of years, ultimately there can be substantial salt accumulation over 5 or more years that could seriously damage the health of a vineyard or orchard.
Okay, thanks, Ian. So another question from Katarina from Huntenburg, our PhD student here at LEC. She is interested in, well, I think it's one of the main problems, I think, to the commercial use, to the practical use of PRD. What about the economic viability of using PRD? Because the second line of drip irrigation can be costly, and what about the ratio between increasing yield or increasing revenue and increasing costs?
Yeah, certainly that's been one of the major concerns with the application of partial root zone drying in drip irrigated crops, the additional expense of installing drip tapes. I think the answer to this depends very much on whether the grower actually gets substantial yield benefits of partial root zone drying. Pascual Romero may be on the call, and he might want to comment further on this, but he did a very nice analysis, cost-benefit analysis of different deficit irrigation techniques in grapevine from some of his experiments in Spain. And if growers were being paid simply by the amount of grape that was being produced, and there was no significant yield increment of partial root zone drying, certainly the investment costs of applying it were simply not worthwhile. If the irrigation, the cost of irrigation water is relatively cheap, again, you're not going to be able to recover additional benefits from saving water by applying partial root zone drying. I put this point to a commercial grower in Washington State who I've got a slide of towards the end of my talk, and his view was that the benefits he was getting with partial root zone drying in his particular wines more than compensated for the costs of the additional drip tape that he had invested in. Again, this seemed to be a sort of a whole package response in the sense that he was applying lots of different treatments, such as cover crops within interrows, as well as his partial root zone drying. But his view was that with the partial root zone drying, he got much better disease control than other growers around his particular region, and so it was valuable. From that perspective.
So I think, Ian, there's another question from Sven. Well, actually there is two questions. One that I left. Well, it was done before, and I tried to go through all the questions from other people. But I think this is I can merge these two questions from Sven, because I think it's quite relevant about the application of PRD in the real world. So Sven is interested in understand if PRD can work if there is a drought, which to me is quite interesting, because I always assume that deficit irrigation can work only if there is a drought, because there is for me this irrigation techniques works to save water. Well, it might not be the target for the grower. So the question is if and he made another comment, if the soil is very dry, would it not better to give both sides a little bit of water rather than have one very dry soil, a very dry side of the soil, and the other a little bit wet? I think probably that's a comment on the interaction between PRD and the volume of water that you are applying, and actually how you can save water. What is the role of PRD in saving water, which maybe sometimes cannot be so straightforward for a farmer if there is no water limitation or no limitation in the access to water. Yeah,
so certainly, based on a sort of scientific analysis of signaling at different levels of water availability, the effect you will get due to partial root zone drying will depend very much on the total access to water that the plant actually has. I am aware that some of the work in South Australia, they were concerned more generally with the impacts of deficit irrigation during a drought, particularly in heat wave conditions. So there was concern that with plants already showing some stomatal closure, if a heat wave came through, that some of the leaves and berries were experiencing such high temperatures that ultimately grape development was compromised. So I think that the partial root zone drying effect will be dependent on the prevailing environmental conditions. I think it's probably quite difficult to predict exactly what the impact's going to be, depending on the level of drought that is experienced over the course of the growing season. Are you still there, Jaime?
Sorry, I mute my microphone. Yeah, so there's I think a final question from Ali Sagnasari. So how do you see further or the future of PRD application in the field, and what happens when we have salinity?
Yeah, I think with salinity, there's always going to be substantial demonstration and testing needed to be done within a research context before growers are likely to apply partial root zone drying or deficit irrigation with any low quality water, because of the risks of improper management resulting in crop losses. Having said that, I know that particularly those within the Middle East, there are whole institutes devoted to devising techniques for growers that have only got access to low quality water. So from that perspective, partial root zone drying could be part of the mixture of a range of different management options, but ultimately it's that long-term sustainability that's absolutely critical, and that will require evaluations over a number of years. And I wouldn't want to sort of downplay the challenges of convincing growers that any particular irrigation technique is going to be viable over the entire lifespan of the crop.
Okay, I think there are no more questions, so you can continue with the presentation.
Okay, well, it's good to see so many questions, and I hope I've managed to provide some satisfactory answers to them. I'm going to move to sort of a more technical part of the talk next. So I guess in the first two parts of the talk, I've sort of outlined some of the scientific theory that underpins the development of partial root zone drying. Then I've looked very much at the agronomy in terms of the sort of global analysis of partial root zone drying effects. And I think that discussion has sort of established that there's still considerable empiricism in how we apply partial root zone drying in field environments. Questions like how often should the wet and dry sides of the root zone be alternated? What are some of the issues that need to be considered with the application of low quality irrigation water? What I want to do now is sort of take a step back into the laboratory environment and think about this from a sort of scientific perspective. And one of the things that always challenged me about partial root zone drying when I first heard it being described was this idea that the signaling from the root system could maintain stomatal closure even as the soil was getting drier and drier, because it seemed counterintuitive that you were trying to promote something coming out of the root system at a time when the water fluxes from that root system will be decreasing. And so I became, captive when I learned that I was going back to Lancaster to do work on partial root zone drying. I became sort of obsessed by the idea of trying to understand how much signal was traveling from each part of the root system. And it took me a number of years to work out how to actually do this. And I'll show you the plant that allowed me to do this in the next slide. But I sort of ultimately developed a technique that allowed me to measure sap fluxes through different parts of the root system, and then subsequently measure the signaling from different parts of the root system. And I developed a very strange plant using a grafting technique called a two-legged sunflower plant, and you'll see the reason for the descriptor in the next slide. The slide that I put up at the moment shows the sap flow through two different parts of the root system. And you can see my colleague, Gregorio Aguirre, who was a visiting PhD student at the time, downloading the sap flow data from this sunflower plant. Now, there were sap flow gauges attached to two parts of the root system. So for the root system that was exposed to wet soil, is indicated by the blue line, and you can see that it was initially high sap flow rate. At night time, the sap flow rate decreased. These fluctuations represent heating and cooling cycles in the controlled environment. Lights come on in the morning, sap flow goes up again, as you'd expect. These arrows represent when the wet side of the partial root zone drying plants was irrigated. And you can see for the roots in wet soil, the sap flow was maintained throughout this experiment. For the plants grown for the root system that was in dry soil, you can see here that the sap flow declined over the period of the experiment. And at this point here, I ultimately harvested the plant to determine the levels of a putative signal, ABA, within different parts coming from different parts of the root system. So you can see here, this is one of these two-legged sunflower plants. So this system allowed me to measure sap flow from either the wet or the dry parts of the root system using sap flow gauges, but also to measure the ABA concentration moving from each root system to the shoot of the plant. Now, I was also able to remove a leaf, measure its water potential using a Schollander pressure chamber, and then collect some xylem sap from that particular leaf. And this is just, summarized the data resulting from these measurements of xylem ABA concentration. So again, we have two treatments, deficit irrigated plants and plants exposed to partial root zone drying with distinct wet and dry parts of the root system. So this is the ABA concentration in the xylem of the leaves, plotted as a function of either leaf water potential and whole pot soil water content. So the ABA is important in partially closing the stomata, restricting water use by the crop. And you can see, although the data are scattered with each point representing a different plant, that there was a different relationship between ABA and leaf water potential, but also ABA and the soil water content of the whole pot. And it's this last point I want to sort of concentrate on, that initially as the soil dried in part of the root system, the ABA concentration seemed to increase, and then later it decreased as the soil in the dry side of the pot got really very dry indeed. Now, contrast that with the plants exposed to deficit irrigation, where initially there was the ABA was relatively stable and then substantially increased as the whole pot dried out on both sides of the pot. So there seemed to be some difference in the actual signaling between the roots and the shoots as part of the root system dried in the partial root zone drying plants. And it's this decrease in xylem ABA concentration of the partial root zone drying plants as the soil gets really dry that I think is potentially very important. So early during the drying cycle, you get a stimulation of ABA signaling, and you can imagine this could close the stomata. But then later on, as the dry side gets too dry, you get a restriction of ABA transport from the roots to the shoots. Now, under these conditions, the plant essentially behaves as a well-watered plant, even though part of the soil is very dry indeed. And so if that's the case, the plants might be able to sustain stomatal opening, sustain photosynthesis, even as the soil gets considerably dry. Now, it's this reduction in stomatal conductance, a reduction in the effect of the signaling that has prompted this suggestion to alternate the wet and dry parts of the root system. And certainly, you know, when studies have applied either fixed or alternate partial root zone drying, there often seems to be a benefit of the alternating the wetting and drying parts of the root system. Again, this is a meta-analysis conducted from studies in the literature, the bulk of which have been conducted in China, comparing alternate wetting and drying plants to fixed partial root zone drying plants. And you can see that for these plants exposed to the alternate partial root zone drying, where the wet and dry sides were regularly swapped, much higher yields than those for whom the wetting and drying parts of the root system stayed the same. And so the partial root zone drying was fixed. So, you know, this comes back to the sort of point made in that internet discussion about whether wetting and drying parts of the root system was necessary, and whether there was anything magic about this. Now, I wouldn't say magic because I'm not a great believer in the concept as it comes to science, but certainly there was a physiological or agronomic impact going on here, and it became important to try and understand what this actually was. So moving back into the greenhouse with some split root tomato plants, we did an experiment where we simply quantified the biomass partitioning amongst different parts of the plant. And at the end of the first soil drying cycle, for both the well-watered plants and the partial root zone drying plants, now these groups of plants received the same irrigation volumes. The plants, the root part of the root system that was in drying soil didn't result in any substantial increment in root biomass. When the wet and dry parts of the root system were alternated, so this wet side was allowed to dry the soil, this dry side was then rewatered, we showed a substantial increase in root biomass in the part of the root system that had been originally dried. Now, over a series of wetting and drying cycles, this effect became quite significant, so that after 4 wetting and drying cycles, the partial root zone drying plants had much greater root biomass than the plants that were exposed to well-watered conditions. Now, within tomato crop grown in greenhouse conditions with sufficient water and nutrients, you'd argue that this root biomass increment is not likely to be of much advantage to the plant. However, if you're in a field environment, actually promoting the root growth could be, you know, really a significant effect, allowing the plant to access water resources deeper in the soil profile if they're available, and also potentially nutrients within the soil. So this rewatering of roots that had been dry seemed to be quite an important response of plants to partial root zone drying. So returning to the effect of alternation on a short-term effect, looking at the ABA concentration in the xylem again. So 3 groups of plants in this particular experiment, with the alternation event occurring at 10 o'clock in the morning in a controlled environment. So we have well-watered plants irrigated on both sides, partial root zone drying plants that have just had the wet and dry sides alternated, and then partial root zone drying plants where the wet and the dry sides have been the same for a week prior to this alternation event occurring for these plants. So soil water content of the 2 groups of partial root zone drying plants was pretty much the same. The soil water contents just differed in terms of which sides were wet and which sides were dry. The leaf water potential of the partial root zone drying plants was less over the course of the day, and perhaps towards the end of the day some separation between the 2 groups of plants. What was noticeable was that when you alternated the wet and dry parts of the root system, you see a substantial increase in ABA concentration following the alternation event. So this alternation of the wet and dry parts of the root system seemed to be very important in actually flushing ABA out of those previously dry roots and into the shoots of the plants. Now, this original work that we did back, in the early 2000s has been a little difficult to reproduce. So I know Juan Gabriel Perez, who was much more careful in actually keeping the wet side of the root system thoroughly irrigated throughout the partial root zone drying period, has struggled to reproduce this effect of partial root zone drying. What he has found, however, is that a substantial increment in ethylene production by the plants if they're subjected to a swapping of the wetting and drying parts of the root system. So you can see the base of his partial root zone drying plants here, grown in our controlled environment room. We have a divider between the 2 different parts of the pot, have some soil moisture sensors measuring the wet and dry parts of the pot, and then by either removing the leaf to enclose in a vial, we can analyze the headspace gas to measure ethylene production, or we can collect the leaf and then measure the concentration of the precursor of the plant hormone ethylene, or collect xylem sap. We see for these partial root zone drying plants that have had the wet and dry parts of the root system alternated, substantial increases in the ethylene precursor, in the xylem sap, in the leaves, and also increased ethylene production by the wetting and drying parts of the root system being alternated. This is our photoacoustic laser spectroscope that Jaime has done fantastic work in keeping it operational to actually measure ethylene production by the plants exposed to the different irrigation treatments. So Juan Gabriel has published this paper recently, and I think this is quite an interesting paper because in his hands, this effect on ethylene production by the plants was so much greater than any impact that he could detect in terms of ABA production, showing that partial root zone drying does have multiple impacts on plant hormones. Now, ethylene is probably most well known to you as a plant hormone that's important in fruit ripening in certain crops, and I'm aware from the literature that some have reported increased fruit ethylene production by plants exposed to partial root zone drying. And I'm also aware that in some cases, fruit harvest date has been substantially advanced by applying partial root zone drying. And I think this observation on ethylene is potentially quite important in trying to understand why you might get changes in fruit harvest date in plants exposed to partial root zone drying. So some conclusions from a more sort of scientific look at partial root zone drying again. Certainly having distinct wet and dry parts of the root system affects the hormone responses of the plant. And actually the alternation of the wet and dry parts of the root system affected hormone responses. It also affected root growth, and this might be particularly important in field environments to allow the grower to apply less water if the roots can access water that is lower in the soil profile, and that there seem to be multiple hormone effects as a result of applying partial root zone drying. So I just need some irrigation briefly, Jaime, but I'll see whether any questions on this part of the talk.
Okay, we have one question so far. So Juan Miguel from Febas is interested in the reasons why in that figure you show with the ethylene evolution from the different treatments, why PRD alternated plants are more variable, so the error bars are bigger.
Yeah, that's an interesting result. I think I'm going to have to ask Juan Gabriel to perhaps explain that for me. Possibly one answer that springs to mind is that, at least within the tomato plant, it is known to be very sectorial in the sense that roots from one part of the roots in one part of the root system are actually plumbed to leaves, to distinct leaves within the canopy. So it's possible that depending on which particular roots are feeding which particular leaves, that the impact of ACC, the ethylene precursor, going from the roots to the shoots may be substantially increased in response to rewatering, or not affected very much, depending on where that particular leaf is getting its supply of ethylene precursor from.
Well, maybe I can step in because I have some experience with this method of ethylene measurement, and it is true that when you don't have an ethylene response, error bars are normally small, because the peaks of ethylene, or the accumulation of ethylene, is very similar across the different leaves in the treatment. But when you have a response, then this is not a really homogeneous response. Some plants, for some reason, are not responding. Maybe for the reasons Ian is pointing out, but also you can see that also in some, when you have a genotypic response, is not that you have genotypic response for all of them. It might have to do something with the transient nature of the ethylene evolution. Some plants, maybe you don't really pick the ethylene increase because it occurs earlier or later, or you don't see for another reason. But maybe Juan can comment further on that.
If there are no further questions, Jaime, on this particular part, I really should get to the last part of the talk, which specifically looks at the commercial application of partial root zone drying.
Yeah, okay, there is no more questions, so you can go on. Okay,
so the last part of the talk is very much focused on commercial application. So you remember, if you're here earlier in the afternoon, I had a map of the globe that was heavily populated by stars indicating trials of partial root zone drying in field environments by academic researchers. Over the last 6 months, I've been contacting a number of you, and I suspect many of you might be on this call because of my recent contacts with you, trying to understand whether the research that has been done on partial root zone drying has actually been applied by commercial growers in the field. And I'm very pleased that Sven's on the call today, because he may be the only commercial grower actually on this call, which suggests that as a scientific community, we still have a lot of work to do in terms of engaging with growers and supporting them if they're interested in trying out this technology, which I think has had a lot of positive reports in academic articles, but is at the moment seems to be relatively not relatively widely applied throughout the world, with one possible exception, and that exception is China. So this is my understanding of where growers in the world are actually applying commercially partial root zone drying at the moment. And if you look at this snapshot of crops and parts of the world where it's being applied, you will see that there's a lot of blank space on this particular slide, and that I've really struggled to find actual commercial application of partial root zone drying, and that is distinct from what I would call supervised trials, where researchers talk to a grower, conduct research under commercial conditions for the duration of a funding cycle, and then report the work in the academic literature, and then lose contact with the individual grower, and there's no follow-up, and they cannot be certain that the grower has continued to apply the technique after the duration of the funding cycle. I mentioned there's some exceptions. So certainly my colleagues in China inform me that there are many examples of farmers applying techniques of partial root zone drying. The technique is usually referred to as alternate partial root zone furrow irrigation, and it's this simplicity of application in furrow crops that I think has been very important in terms of the commercial uptake of the technique. Obviously, we discussed earlier that drip tape is expensive in drip irrigated crops, and that might be a significant constraint to the application of partial root zone drying. In furrow irrigation, actually, if fewer furrows are being opened to irrigate the crop, if they've been already constructed, this could represent a considerable labour saving for the farmer. So my colleagues in China assure me that the technique's been applied in a number of different crops, and that farmers are actually using it. And you can see here an example of vines grown in, I think, Gansu province in northwest China, where I know my colleagues at China Agricultural University have done a huge amount of work in trying to preserve regional water resources over the last 20 to 30 years. And you can see here that, for your conventional furrow irrigation, where every furrow is irrigated, you can save a substantial amount of water if only every second furrow is irrigated. And I'd encourage you to have a look at the work of Taisheng Du and his colleagues who've done some nice work with this particular technique with alternate furrow irrigation. In the rest of the world, I think it's a bit more difficult to find examples of where partial root zone drying has been applied. There was some nice work by Felipe Barrios Macias, and I apologize if I've mutilated your name, Felipe, on some processing tomatoes that were subjected to furrow irrigation in California, and they published this work a number of years ago, showing some quite positive responses. So in this case, this seemed to be a direct and in some cases transient response to water scarcity within an irrigation farming community. So in this particular case, the alternate furrow irrigation, which is analogous to partial root zone drying, was applied during the sort of peak stress of the California drought, and also as a result of the research project that Felipe and his colleagues were working with. I understand from talking to extension agents in California in more recent times that actually the bulk of the industry has now moved away from furrow irrigation to much more water use efficient drip irrigation, which is obviously great for the industry in terms of conserving a very scarce water resource in that part of the world. But for partial root zone drying enthusiasts such as myself, it's perhaps more difficult to satisfy my need to know whether these techniques are being commercially applied by farmers. One interesting little aside, within the SHui project, we have a remote sensing group, and I'm not sure whether they're on the call this afternoon or not, but certainly one of the challenges, I think, in documenting the application of partial root zone drying within a sort of wider area like California is just to understand, which growers are applying it and how often they might be applying partial root zone drying. Whether we could use remote sensing to actually determine whether growers are applying partial root zone drying or not, I think is an interesting research question. So you can see with this image here, taken from some of Felipe's work, that you can clearly see a furrow that has been irrigated and then one that hasn't. Now, whether this is viewable from space, and we could use remote sensing to determine how widely used the technique is across a region of the world, I think is an interesting technical but also intellectual challenge to understand how widespread this technique has actually been applied. One other example from the United States is from Washington State. I talked about Paul and Barbara's vineyard earlier. I've had the good fortune of having a talk to him thanks to the marvels of modern technology, where he was out in his vineyard on his phone showing me exactly the extent of their operation. Very beautiful vineyard in a very beautiful part of the world with snow-capped mountains of the Cascades in the background. For him, partial root zone drying is being applied as a part of a wider series of innovations, including the use of cover crops and various other organic farming techniques, and certainly reduction in pesticide application, particularly for mildew, has been one of the benefits he has had on his particular vineyard. I think it's perhaps a bit more difficult to understand, given that he's invested in partial root zone drying throughout his operation, whether this is a general benefit of partial root zone drying or whether there's synergies with some of the other techniques that he has applied. Certainly, he's very much an enthusiast about the technique, and as far as he's concerned, in terms of the partial root zone drying, it paid for itself within the first year as a result of much better mildew control compared to his neighbours that were applying conventional irrigation techniques. And you can see here how he's split the irrigation tapes to ensure that he can apply his partial root zone drying. One final example of commercial application, and I'm very pleased to have you on the call, Sven, and I look forward to meeting you at some point, is your vineyard in Chile, which again I only became aware of while I was preparing my slides for this afternoon's presentation. Obviously a much drier looking environment, and I think this has been borne out in your questions. And I think this is a really nice example of a substantial investment in partial root zone drying, with installation on more than 20 hectares, following a quite close collaboration with a local university. And I think this provides a successful model for those of us who are academics and researchers on partial root zone drying, to actually gain some satisfaction that the work that we are doing from an academic perspective can actually be applied in the real world. So certainly I'm aware from a previous European Union project of farmers collaborating with academics in the irrigation science community. And it's often commonplace in such trials to say to the farmer, well, look, we'd like to work on your farm. Please, can you just continue to irrigate as you normally have done, and we'll set up our trial and do our own forms of irrigation that we are interested in. And certainly I'm aware from a previous project that didn't use partial root zone drying, were actually because of the interaction between the farmer and the academic, that the farmer irrigation scheduling changed over time so that much less water was actually being applied once the academics had been able to demonstrate that the irrigation scheduling they were doing was able to save water without any compromising of the quality of the fruit that was being produced. So I think this idea of stronger collaboration between the grower community and the academic community is absolutely essential if techniques such as partial root zone drying are to be widely applied. I say this as a considerable caveat given the earlier meta-analyses I presented, where it seemed to be that relatively few academic trials of partial root zone drying in field environments resulted in substantial, more than 20% yield increments. So possibly some of the commercial examples I've provided over the last few slides are these sort of outlier points where a particular case of variety, soil type, climate has resulted in conditions where partial root zone drying has resulted in a substantial increment that's of commercial significance. So certainly I think that there is some scope for further work in this area to understand the conditions under which partial root zone drying can offer these benefits to growers. So to sort of conclude the sort of more field or commercial evaluation of partial root zone drying, certainly I've found it difficult to find actual commercial applications outside of China, where I understand the technique is widely being applied. Those that I have found, often the growers are very enthusiastic about partial root zone drying, are often highly educated or supplying niche markets. Having said that, given that furrow irrigation is still relatively widely applied in some parts of the world because of the lack of infrastructure investment in drip irrigation or other forms of localized irrigation, I think alternate furrow irrigation or partial root zone drying is a technique that can be widely applied if the advantages or benefits can be adequately demonstrated to growers. So whereas there are we've talked a lot this afternoon about science and some of the sensors that could be used to perhaps more optimally apply partial root zone drying in specific situations, the reality is that the empirical and I would say legacy approach of furrow irrigation may well provide environments where partial root zone drying can be much more extensively tested. So I think there is still some work to do by the academic community, particularly those of us that are interested in working in a development context. So I would argue there's a lot more work to do. I'm obviously very happy to take questions at this point and interact more with you. I should just say some thank yous. First to my colleagues in the SHui project that sort of I've interacted with over the last couple of years and sort of put me up to this in some extent in the sense that I've asked others to provide webinars on topics, so it seemed only reasonable that I choose a topic that I wanted to talk about, and hopefully I've communicated some of my enthusiasm for partial root zone drying over the course of this afternoon. I would like to thank the expanding, and I say that in question marks, partial root zone drying community. It was a topic that I've not really considered much over the last 5 years up until a couple of recent projects. And when I went back to the literature, I was amazed at how many new research papers had been published with this particular technique. And obviously, thank you to those who have patiently fielded my inquiries about my obsession as to whether this technique is being widely applied within a commercial context. I suspect that partial root zone drying or deficit irrigation more generally is only going to be widely applied in very arid environments. And so it's no coincidence that a lot of my colleagues over the years working on partial root zone drying have been Spanish. And so thank you for your support of my academic interests, and also for the various samples of wine when I've been actually able to get to Spain to collaborate in field trials. And just a couple of other people that have been very influential to me over the course of my interest in partial root zone drying. Professor Bill Davis, whose work I started off citing, looking at laboratory split root experiments, and I think it's been a really nice example of how a fundamental question has actually been taken up by not just the wider academic community, but also the grower community as well. Also, Dr. Brian Loveys from CSIRO plant industry in Australia. He has his own vineyard now, and I think he's still applying partial root zone drying on that particular vineyard. So I would just close with one final anecdotal story, and that is when I returned to Bill's lab to work on partial root zone drying at the start of this century, and I was talking to my father about partial root zone drying and the experiments I was hoping to do with it, and he said, well, I can remember someone talking about that at an international horticultural congress that I went to in 1980 in Australia. It was a guy from the Victorian Department of Agriculture, and I said, oh well, if you can find the information, I'll be very pleased to receive it, because it's a technique that I'll be doing some research on, and clearly it's valuable to understand what comes before any research that I might do now. So my father rang up a lot of people in the Department of Agriculture in Victoria to try and understand who had done the work and whether any records had been kept, and his search, which was quite extensive, turned fruitless. He was unable to find any evidence that work had been done, even though he distinctly remembered going to a talk about it at an international horticultural congress in 1980. And so if you're a student who's listening to this presentation, I think this places importance on the benefit of publishing. That ultimately, if your research, no matter what the conclusions are, isn't captured by the academic community in the form of a publication, there's actually no official record of the work having been done. So at this point, I will re-irrigate myself, and I'm very happy to take questions. If you've been with us this afternoon, thank you for your attention. I'm pleased to see a lot of people have been on the call this afternoon. Very happy to take questions at this point.
Yeah, we have a comment from Bill. So Bill is making this question. So the meta-analysis suggests that the somewhat limited extra scope about DI for increasing water productivity. If we think about using soil drying as a growth regulator more generally, are there wins here? For example, speeding up development, early harvest of cotton, enhanced ripening, and maybe most important, product quality. For instance, some Portuguese work on wine quality.
Yes, certainly there are a number of benefits of partial root zone drying that I haven't really considered this afternoon, and so I'm very pleased that you've highlighted some of those, Bill. Yeah, there's I think the water saving aspect of it is only likely to be of relevance to growers when the costs of buying in water are going to be substantial. It's the improved quality, and I've only briefly alluded to that this afternoon, that is likely to persuade growers that this is a useful technique, and certainly there's been a large number of scientific papers published over the last 10 years showing improved quality of different crops in citrus, in grapevines, I think in apple as well. And having said that, though, I still query whether these are the outlier studies or not. The meta-analysis that Michael Adu did recently suggested very limited impacts of partial root zone drying on quality. But again, this was a relatively limited perspective on quality, just looking at total soluble sugars. And that for some of the crops we're interested in, the measuring quality is a lot more complex. And, a lot of, quite detailed measurements need to be done to actually understand the impact on, say, grape berry chemistry, to understand what the impacts of partial root zone drying might be.
Yeah, so just a couple of questions from Sven. So how big is the Oxford Landing vineyard which they apply PRD? And another question that maybe Alon, I think, joined us earlier. Why is not used in Israel?
So I'll answer the first one. The Oxford Landing vineyard is, they only apply the partial root zone drying on one block of one of their vineyards. At least that was the case in, I think it was 2006 when I visited. So they were using it just on one block with one specific variety, and I think this was largely a legacy of a previous trial that they'd done with the CSIRO plant industry. It hadn't been applied throughout their operation. If Alon is on the call, and I know I saw him there earlier, do you want to answer Sven's question about the application of partial root zone drying or otherwise in Israel? Perhaps he's not with us.
Yeah. Okay, so there is another comment from Felipe Barrios. So even without a significant increase in yield, he considered that water saving capacity, more than 25% without a yield decrease, is already an encouraging benefit to deal with future droughts and food security issues. And I think that's an interesting question, because we might have here the controversy between the farmers wanting to always have water available. And so I know that in some regions, for instance in Spain, they don't want to hear about these kind of things, because they want the government to provide enough water and not thinking of saving water, and the real need of saving water at some point in the future with climate change.
Yeah, I think Felipe makes a very valid point that is going to be determined entirely by the by the water scarcity within the prevailing environment, and whether regional governments, because it's usually regional governments that are involved in this kind of provision of infrastructure, are able to spend money to boost water resources in the area. At least in a UK context, and this might surprise you, Felipe, to know this, there's actually a substantial benefit of water saving for some industries that are looking to expand their businesses. And the reason for that is that each particular business has to apply for an irrigation license within the UK. And the irrigation license is capped now, so that if you're a particular business, you can continue to apply for that license, but you cannot increase the amount of water that you can extract on that particular license. So what that means is that if you're a very profitable grower and you're irrigating your crop, and I know probably the most profitable crop that's grown in the southeast of the UK, which is one of the more water scarce regions, not just for climatic reasons, but also because there's a large population close to London in Kent, which used to be which was still regarded as the sort of fruit basket of the UK. Berry growers will have their berry crops, strawberries, raspberries, grown in polytunnels. They will harvest rainwater to some extent, but they'll also have an irrigation license. Now, as the demand for berry crops has substantially increased within the UK, a lot of these growers have looked to increase the size of their business because they know that they can sell more berries if they can produce them. The challenge for them is that they can't extract any more water than what they were doing 20 years ago when their business was much smaller. So what that means is that if they're going to increase their production, they need to be much more efficient in how they use the water that they are allocated. And certainly one of Bill Davis's former students, Mark Ellis, has done fantastic work with the berry industry here in the UK, improving their water use efficiency, not just using techniques such as partial root zone drying. It seems as though partial root zone drying can be a little problematic to implement in some of the new growing substrates that the growers are using in containerized crops. But certainly, the water saving can be a substantial benefit in particular regions if it allows business development opportunities.
So Pilar Gil from the Universidad Católica de Chile is commenting about that. So in Chile, it might be that water saving techniques will be mandatory in the future because they have been facing a terrible drought in the last 10 years. So that could be the most important issue, the most important benefit for PRD and other techniques. Also, she's asking a question about whether PRD could affect negatively to the fruit conditions. So the increase in ethylene could affect negatively to the fruit or provoke any kind of physiological disorders.
Yeah, I'm not a fruit physiology specialist, so I'm not so certain about disorders. I would argue that if you're increasing fruit ethylene production, you may accelerate ripening. Whether that is beneficial or not, I think will depend very much on the market that you are working in. I know from talking to Juan Gabriel Perez working in Spain with his trials on partial root zone drying in lemon, that the crop comes in much earlier and so consequently attracts a higher price within the marketplace if the lemons can be harvested earlier. There may be situations where an earlier crop is not what you're trying to achieve within your growing environment.
Yes, she's asking that because as Chile is an exporter of fresh fruit, so it might have an impact on the post-harvest fate of the fruit, or what happened with this of the fruits.
Yeah. Again, I think there are a few studies starting to emerge on partial root zone drying impacts on post-harvest handling quality of crops. In fact, Charo Conesa, who's working at University of Cartagena, Polytechnical University of Cartagena in Spain, has done some work on partial root zone drying and post-harvest quality. I'm going to have to direct you to her papers because I can't quite remember what the results were. But certainly, it's an area that needs additional attention, given that for export, that's obviously a principal determinant of the economic value of the crop that's produced.
Okay, so just a final comment from Juan Gabriel Perez in line with the comment from Bill. So he agrees on the value of PRD to manipulate the quality, and I think in his case, because I have been talking about this with him while he was here, I think one of the benefits he has highlighted or he has seen in his trials is the possibility to anticipate the harvest or having a harvest in an early harvest, which can make a massive impact on the economy of the farmer. So by, for instance, changing the root to shoot ratio, he has seen that there is an anticipation of the having fruits with the right caliber in an early part of the season, which is making a big impact. We have another comment from Bill.
Yeah.
So the comment is from one of the big benefits seen by the industry from your work and that of others in water scarce regions is that growers gain a lot just by metering water and measuring soil moisture status. So by seeing what the academics are doing in terms of measuring how much water they are putting in and measuring how much moist is the soil, they can just gain a lot.
Yes, certainly. I think much better collaboration between academics and growers is going to be essential to deliver improved water use efficiency in water scarce regions. Now, whether growers decide to adopt partial root zone drying or not, I think is to some extent a moot point unless they can see real tangible commercial benefits of applying the technique. Just having a much better understanding of the water requirements of their crop and measuring that is going to be increasingly valuable, particularly as I think some governments around the world are going to be forced into charging more for the water that is available. And I think as a community, we need to have a much better appreciation of the value of water in arid regions, particularly given that agriculture is increasingly required to show its environmental credentials. So any discussion amongst the academic community that stimulates growers to think more carefully about the decisions they're making on irrigation can only be welcomed.
Okay, so I think there is no more comments. Well, on behalf of the many comments we are getting, thanking you for the presentation. Thanks, Ian. It was really a very insightful presentation and a very nice summary of the state of the art about PhD, and we hope more people will join us as a PhD community in the future.
Yes, so thank you to all for coming along this afternoon. I hope you found something of interest in the presentation to you. Obviously, I'm more than happy to continue discussing offline with you any particular aspects of the presentation, but also just a big thank you to those who I've been discussing with recently, because it's only via collaboration that we can hope to understand, not just the science behind the technique, but also, develop strategies to make similar techniques, much better communicated to interested growers.
Okay, so just one last comment I want to make from Sven. I think he likes the term PhD community. Maybe we can coin this term and make it more popular in the future. PhD community.
Thank you, Sven, for that comment. Yes, I think it is very much a community, and certainly it's a community that I've benefited a lot from over the course of my scientific career. For me, there's been some interesting questions that have been asked by growers, particularly that forced me to think about what I'm trying to do with the scientific experiments that I'm undertaking. And I think it's this interaction between growers and scientists that can only benefit the broader community beyond PRD. Okay, I think at this point I'm going to say farewell to everyone on the call. I hope you found it an interesting afternoon, and like I say, I'm more than happy to discuss with you at any future point in time. Okay, bye for now.
Thanks, bye.